So does anybody have any specific questions besides I don't besides I'm not ready for the test?
Um, I don't know, it just seems like it's very chapter five heavy.
Very
Um,
chapter 5 heavy. Indeed it
yeah.
is. You're correct.
And then yeah, and then just seeming like chapters one through three. Like I'm making some notes, but I feel like it's like, oh, I feel like this is really more anatomy. Like we should know more anatomy on this. So,
There is not any anatomy on the test.
oh, okay, boom.
Like I've said a couple times in the lectures, and whether I don't know if you heard this, but I said this is not a neuroanatomy class. So yes, in chapter 2, it kind of reviews neuroanatomy, and I
Okay.
briefly went over that, just in a way of like you take an overall view of the map before you go into different areas. So I don't expect you to know any of the neuroanatomy. And in fact, I'm not going to ask you any of it except things about like white matter versus gray matter,
Got it.
you
Okay.
know, that sort of thing. So gray matter is the somas, the bodies of the
Right.
neurons, and white matter is the axons. So that
Right.
is the limit of the
Yeah.
neuroanatomy that you need to know. So the other things from chapter 3 specifically was talking about neurological examination, distinguishing whether something is an acute vascular problem or a subacute inflammatory problem or a chronic degenerative problem, that kind of thing, given a brief case study. And then just the purpose of um, doing a neurological screening exam. So a lot of times, in a just a regular PT evaluation, they do not a lot of times, every time they do a neurological screening exam. And what, why? Like someone came in for an orthopedic issue, why would they do a neurological screening exam? Any answers to that question? Any
No, I mean,
ideas?
I think it could be lots of things. I mean, we're, you know, just because it's ortho doesn't mean like we're just fixing, we're rehabilitating a knee, a hip, a shoulder, whatever. But if they have impairments that, say they have Parkinson's or they have, uh, you know, anything that's going to, what's the word I'm looking for?
Impede their progress?
Impede or impair their progress. I think that's good to know instead of like, well, why aren't you doing this? Why aren't you getting better? And then we find out later on, oh, it's because of X. Like, oh, well, crap. If had we known that, we probably would have taken a different direction or approach.
Exactly. And so one of the things is to, well, like I always say, nobody ever comes into the clinic without their nervous system.
Mhm.
So, just like they don't come in, they don't leave their mental health disorders at home either.
Right.
So a neurological screening exam is really to determine if there are any underlying issues that need referral to another practitioner. That's another thing. And so that's a lot of times the PT, when they're doing the initial evaluation, they're not just looking at the current injury or deficits. They're also trying to see it, they're trying to give more comprehensive care. So doing a neurological screening exam allows them to do that and know if there's any underlying neurological disorders and whether or not they need referral to maybe to a neurologist or to somebody else. So that appropriate recognition of problems and referral is a really important part of not just what PTs do, but also PTAs. So we might not be able to treat the issue, but we can certainly refer appropriately.
Gotcha.
So that makes sense. And then chapter 6 um, is a lot about neurotransmitters and the events at the synapse, of course. And we don't need to go into like chemical detail about the neurotransmitters, and what type of substances they are, but just kind of their primary functions. So, what determines whether a neurotransmitter is excitatory or inhibitory?
But HD. You would think.
Mason's giving me the eyebrow.
Oh, I was trying to
Mason,
understand
just
what
raise your
Darren
hand.
said.
Just raise your hand.
So really the effect of a
Type of
neurotransmitter,
transmitter it binds to.
right, it depends on the receptor that it binds to. So if it binds to an inhibitory receptor, it's going to be inhibitory. If it binds to an excitatory, it's going to be excitatory. So the effect of a neurotransmitter always depends on the receptor that it binds to. So acetylcholine has one effect or many effects in the autonomic nervous system, and it has a different effect at the neuromuscular junction. So its effect depends on where it binds. So the receptor that it binds to, and then in chapter 7, talking about neuroplasticity, that is one of the ways that a neuron can change its function is by having different receptors. So the receptors they're proteins. They're coded for by the DNA. So if you're getting a certain type of neural input, that can impress upon the cell that it needs to make different receptors because something else is going on. So that's kind of the main thing with neuroplasticity is making the changes in response to whatever the current setting is. So when we do we do vigorous exercise with people with Parkinson's, we're trying to drive neuroplasticity. In other words, we're trying to stimulate the cells to create receptors for the dopamine you do have, or other dopamine agonists, or the medications that you're taking, so you're getting better use of the dopamine in your system because you have a limited amount of dopamine. There's also research that supports that if you like vigorous exercise causes dopamine to persist longer in the synaptic cleft before it's reuptaken into the cell, and so you get better use out of the neural resources that you do have. So part of the neuron is able to change in response to what's currently going on. So that's the whole idea of neuroplasticity. And when we talked about, like last quarter, when we talked about accommodation to electrical stimulus, that's really habituation. That's diminished response to repeated benign stimulus. So you get used to it, basically. Habituation is just a fancy way of saying getting used to it, but it's the neural version of getting used to it. So your neurons stop responding to something that keeps happening that's not hurting you, basically. So I always say I don't know, I don't think I used this example in the lecture, but a long time ago, I lived in a place that was really close to the railroad tracks. And so you'd hear the train go by at a pretty regular time every day, and you could hear it. I mean, it was real obvious. And people would come over, like my friends would come and visit, and they'd say, "Wow, doesn't that bother you?" I'm like, "Doesn't what bother me?" You know, and the train, because I was used to it, it was a repeated benign stimulus. I just stopped listening to it anymore. At first, I think when I moved in there, it probably bothered me, but or like you move in some place and it smells different, and you have trouble sleeping or something until you bake cookies, and then it smells more like home, or it smells more like Tony's mom's home. Oh, there's the tuxedo cat.
Yeah.
He's like, okay, right there. That's the spot.
Yeah.
So, with with regard to neuroplasticity, we also our neurons need to change in order for us to learn things. So, that's why I'm always going on about how cramming doesn't work, because you don't have time for your neurons to actually encode the information into long-term memory. So if you just look at it the night before the test, Mason, I'm teasing you because you said you weren't ready. If you just look at it the night before the test, then your neurons don't have a chance. You're just not giving your neurons a chance. So if you do spaced retrieval over time, then it gives you time to encode that into long-term memory and gives your neurons a chance to take in that new information. So neuroplasticity and learning is a neuroplasticity practice, if you want to think of it that way. So anything we learn. So what other questions do people have? So you've been making your cheat sheet,
Working on it.
Working on it.
working on
Final.
it. So what do you have on your cheat sheet?
Yeah.
Anybody want to give us an example of information you have on your cheat sheet?
I don't think anything I have is really valid. I've just been
Yeah.
making notes about
Well, that's unfortunate. That's a waste of a
what
perfectly good piece of paper.
I know. I was rewatching, going over study guide questions and kind of highlighting things, and I'm like, well, maybe I need to know this. Well, maybe I need to know this, but
How about parts of a neuron? So give me
like
some parts of a neuron and their function. That'd be a good thing to have on
I got that.
your sheet. You've got that. That's good.
Yep. I type it all up so I kind of can lay it out, move it around, then I handwrite it.
Oh, that's a good way to do it.
So
I like that.
otherwise I run out of space and it looks messy.
Doesn't matter how it looks as long as you can read it. So you don't get into the issue where you're trying to write so much stuff that you can't read the micro printing. There's one student in one of my other classes who has the tiniest handwriting I've ever seen in my life, and it's also extremely neat and legible. So I'm like, wow. I mean, it's like a 5-point font. I mean, it's really small, but and very, very neat. But so the parts of a neuron, the soma is the body of the neuron. That's where all the cellular function goes on. Axon hillock is the projection that goes into the axon. Axon is the part that takes information to other cells. The dendrites are receivers. They're receiving information from other cells. You have the end of the axon where it meets another cell is the presynaptic terminal. That's where the synaptic vesicles are released. The part of the next cell that receives information is the postsynaptic terminal. So it could be another neuron, it could be a muscle cell or a gland, some kind of effector cell in the autonomic nervous system or something like that. So if it's another neuron, the synapse could be the axon onto a dendrite of another neuron. The axon could be synapsing onto the soma of another neuron or onto the axon of another neuron if you're going to do some presynaptic facilitation or inhibition. So if the first neuron is going to facilitate the propagation of an action potential, it could be synapsing with the axon of the second neuron to say, yeah, take that calcium down to the synaptic cleft, open those, draw those vesicles, get that neurotransmitter going. So, what's going to cause more neurotransmitter to be released?
an excitatory.
Yeah, more excitatory stuff. And then chemically speaking, what's going to happen if you have more excitatory impulses?
Or some more neurotransmitters kick out.
Well, definitely it creates more neurotransmitters, but if you have more calcium,
Oh,
there's
okay.
a greater influx of calcium, which is going to happen if you have more excitatory impulses, then you're going to get more neurotransmitters released. And the neurotransmitter floats, diffuses across the synaptic cleft, binds to receptors. What happens when it binds to a receptor?
It's the key, right? Or that's the way ever since you made the uh, gated community thing, I just can't get that out of my head.
That's right. Either it opens the gate, it
Yeah.
opens a channel,
Mhm.
and allows neurons to diffuse down their concentration gradients, or it has some effect inside the cell. And the effect inside the cell might be stimulating the DNA to create more receptors. If it's something that you're getting a lot of, that synapse is getting used a lot. So most of the time it opens an ion channel. So then you can propagate the action potential down that neuron. So most of the time that's what happens. It binds to the receptor, the receptor changes shape and opens an ion channel. Sodium rushes in, potassium rushes out, everything goes nuts, action potential down to the next neuron, that sort of thing. And then, so what's it called when the sodium rushes in and the potassium rushes out and the membrane potential becomes less negative than its resting potential? What's the name for that?
Just depolarization.
Depolarization. So you have to depolarize the membrane in order to create an action potential. You have to depolarize it sufficiently in order for it to create a potential that's going to propagate. So what's that called? The sufficient amount of stimulation that's going to cause an action potential.
Threshold.
The threshold, that's exactly what it is. So you have a threshold potential, it stimulates an action potential, and it goes on. The ways you can do it is you can have lots of different stimuli, one after another, or you can have a whole bunch at once. So does anybody remember what those two are called? So a whole bunch at once, or they all add up together.
It's evading me right now.
So when they all add up together, it's called summation. Add it all up. And when you have a whole bunch at once, it's spatial summation. So spatial, more space on the neurons being taken up, if you want to think of it that way. If you have one after another, after another, after another, that's temporal summation. So, separate times temporal. So you either have temporal or spatial summation. All of those impulses add up to enough to generate a threshold to generate an action potential. So just like the little terminology things, those are little handy things to know. So does anybody, you know what, I've got to say, with the events at the synapse assignment that you all did, I thought it was really neat how everybody's sort of really tuned into sort of their interests and personality. I thought that was great. You all did a great job at it. And when I read it, I thought, oh, that's so Mason. That's so Darren. That's so Tony. That's so Ryan. That's so Amanda. So Trisha. So it was I was very happy with how well you did that. So
Zach's was fun.
Zach's was fun. He had the whole gridiron thing going on, and he had a video. So he was the only one that did a video this year. So and the funny thing is I when I read people's assignments or watch their videos or whatever, I learn something about them. Um, there was one person who did a video a few years ago who her neurotransmitters were little horses, and somebody did horses this year too. And I like that idea of the little horse galloping across the synaptic cleft sort of thing. But the person that did the video, she actually had like little model horses. I think they were her daughters or something, and she did the video with those. So that was kind of cute. And that's what I was asking Zach if he ate the M&Ms after he did his video. Or I couldn't tell if they were M&Ms or Skittles from looking at the video. They might have been Skittles. So, but I thought that was good. So, in terms of who wants to tell me what excitotoxicity is?
Is that the buildup of the glutamate?
Yep, buildup of glutamate kills the cells around it. So it's the so you have some kind of cell damage, and you have that excess glutamate from the cells that kills the cells around it. Not good, but it happens. Happens when you have neurological damage. So the when somebody what are the things when somebody has central nervous system damage, what are the things that keeps it from healing?
No go.
No go, yep, the neurite growth inhibitor hormone, which is, too bad. Um, Really, when you have something like that that's specifically identified, that can be a target for pharmaceutical applications. But why? Well, I'll start talking about this in chapter 25. But if anybody wants to venture a guess, um, why do um, why do we have trouble getting medications into the brain?
It can't cross the blood-brain barrier
Yeah, the old
sometimes.
BBB. So when they create medications, they have to have something that crosses the blood-brain barrier if they want to affect things in the central nervous system. And that's a challenge because the blood-brain barrier is set up to not let stuff get across. So with dopamine, they just using dopamine didn't work, so they had to use a precursor format, L-DOPA, which can cross the blood-brain barrier and then get converted to dopamine once it's in there. In 2012, some researchers in Japan successfully implanted neural stem cells into somebody's substantia nigra. And then I haven't heard anything about it since, so I don't know what happened with that person. I've been like keeping an eye out, like, heard anything about that? So it's been a while, but so you sometimes there was also a thing I saw years ago where they some surgeons in San Diego had this avascular matrix that they used to replace tendons, and they did surgeries on several people that were successful initially, but then I never heard anything more about it. So I can only imagine that some problem arose afterwards, which is kind of interesting. But so in terms of the questions that are likely to show up on the exam, you always have to ask yourself, now that you know what kind of questions I usually write, it's like, what questions is Loretta likely to write on the question on the test? So what questions do you think I'm likely to write on the test for chapter 5, since there's so many questions for chapter 5?
Guessing something with the 7-step synaptic sequence.
Absolutely, that will be
Just
on there.
shot in the dark.
That is not a shot in the dark. That's a shot in the light. So yeah, and hopefully having done that events at the synapse assignment, it's all crystal clear to you what the 7 steps are. So hopefully you'll know that. What other questions do you think I would be likely to write? Well, we said the parts of the neuron. How about types of neurons?
Like the
Like,
unipolar.
yep, the pseudo-unipolar and the bipolar and the multipolar. Yeah. And where they're likely to be found. What about
Afferent and efferent will be on there.
yes, absolutely. So where do afferent neurons go to and from, and where do efferent neurons go to and from? So that's a good thing to know. What else? What else do you think is likely to be on the exam?
Electrical potentials.
Electrical potential, sure. So resting potential versus action potential, the threshold, in order to, I'm not going to ask
Minus
you,
55.
I'm not going to ask you that. It's just going to say, you don't have to know that it's minus 55 millivolts, that it goes from minus 70 to minus 55. But you do have to know that when the neuron gets depolarized, the membrane potential becomes less negative. And if it's hyperpolarized, it's more negative. So if it's more negative than resting potential, what is the effect? What effect does that have?
Less of a chance of a action potential.
Yes. So it's harder to stimulate the neuron if it's hyperpolarized. If you are more negative than resting potential, it takes a larger, more intense stimulus to stimulate that neuron. And there's even a little bit of an absolute refractory period where the sodium potassium pumps are working really hard to restore things where it's not even possible to depolarize the neuron. Short, but there you go. But there it is. So, what are the main ions that are involved in maintaining that resting membrane potential?
Potassium, sodium, chloride.
Yeah, potassium, sodium, chloride. They're those big fat anions that are trapped inside the cell because they're too big to go through any of the ion channels. Those kind of maintain it more negative on the inside. At rest, it's more negative on the inside and less negative on the outside. That's that minus 70 millivolts difference between the inside and the outside. There's more potassium on the inside of the cell, more sodium on the outside of the cell. How do the sodium potassium pumps work to restore that balance? Or more simply, what do the sodium potassium pumps do?
Is it 3 sodium out, 2 potassium in?
Yeah, 3
That-
sodium out, 2 potassium in. And what's the cost? What's the fee for that?
ATP.
One ATP molecule gets 3 sodium released and 2 potassium captured. So they're 3 sodium out of the cell and 2 potassium into the cell at the cost of 1 molecule of ATP. So every time the pump cycles, it requires 1 molecule of ATP. And then you bust off that phosphorus, you get the energy that's in that chemical bond. Now you have some adenosine floating about, and it binds to receptors that make you sleepy, make you tired because you're running out of energy. And caffeine actually blocks those receptors, which is kind of interesting. So, what do you call a medication or substance that blocks a receptor that causes the neurochemical that would normally bind the receptor not to work?
Would that be considered a stimulant?
Well, caffeine is, but it's not always a stimulant. It depends on what the receptor is.
Inhibitor.
No, but that's a good that's a good thought. I like that thought.
You're looking for
Antagonist. Yes, that's it. And so a medication that blocks the receptor can be an antagonist, like beta blockers in the case of the cardiovascular medications. Those are epinephrine, norepinephrine, and epinephrine antagonists. What's a medication called that enhances the effect of a neurotransmitter?
Again, one more time.
A medication so we said the ones that block the effect of a neurotransmitter are antagonists. What are the ones that enhance the effect of a neurotransmitter?
Would that be an
That one was the-
anti-antagonist?
Agonist.
Close. It's an agonist. Yes. Which is the word for an anti-antagonist. It just takes out the double negative. So an agonist is something that enhances the effect of the neurotransmitter. An antagonist is something that blocks the effect of the neurotransmitter. So a lot of medications are either agonists or antagonists when you're talking about things that treat neurotransmitters. So, how does a neurotransmitter differ from a neuromodulator?
Neurotransmitters are quick and neuromodulators take a long time.
That's one way. What's another way? Or they last longer. And they
Yeah.
do take longer and last longer. Yeah. What's the second way that they differ?
Hmm.
So neurotransmitters are usually released at the synapse, the synaptic cleft, and they just act locally on the next cell in the chain. Neuromodulators are often released in the bloodstream and they act on a lot of different neurons. So a lot of neurological medications also act as neuromodulators in the same way. So things like gabapentin and the things that act to diminish response are usually neuromodulators, and so they act on a lot of different cells at once. Some of our hormones, like norepinephrine and epinephrine, are neuromodulators because they're released in the bloodstream and they act on a bunch of different cells at once, and neurons at once. A neurotransmitter is usually just one cell to another, like in the relay race, passing the baton, and the neuromodulator is all over the place. And neurotransmitters act fast, and the effect goes away fast. And neuromodulators act more slowly, and the effect persists longer. So, what uh, what type of axoplasmic transport takes substances from the soma out to the presynaptic terminal?
So
It's the handle toe at the ski area.
what was the question?
What type of axoplasmic transport takes substances from the soma out to the end of the axon, to the presynaptic?
Anterograde?
That's anterograde transport. And then the synaptic vesicles go back to get refilled by the other
Retrograde?
kind, retrograde. It's axoplasmic transport. And as we get older, or if with degenerative neurological diseases, axoplasmic transport slows down, and so nerve action slows down. So I was doing for my workout today, I was doing like plyometrics and agility training, because I don't want that stuff to slow down any more than it has to, right? So you got to do that stuff, do those fast steps. So then when you start to fall, you can fast step out of it. So let's see. Chapter 8 will be on the next test. That's the development, which is interesting. Um, what? So, how soon after someone has had a stroke do we want to start rehab?
Immediately.
Yeah,
Right?
pretty soon
If possible.
afterwards, 5 to 30 days. That's kind of the optimum window. And what type of training are we usually going to do?
Repetitive.
Biometrics.
What did you say?
Repetitive.
I heard Darren, but then somebody
Oh.
else said something.
Plyometrics.
Oh, probably not plyometrics. People can do plyometrics after a CBA, but functional training. You want to do repetitive functional training. So things that I worked with this one guy who had a stroke, and his wife, she was really clever with coming up with, she got like these plastic cups and she would have them, because we were doing cones and stuff in the clinic, and she'd have him stack the plastic cups, like stack them all over here and then stack them all over here. So she's doing it across the midline, functional. And then she upgraded him to unloading the dishwasher. I'm like, that's a great repetitive functional task. So, The repetitive functional, do it early on. That is high intensity. That is definitely going to be the optimum. But we don't want it to be too. Why don't we want it to be too high intensity?
Because they're just getting back to. I mean,
Yeah, you can actually
it's-
damage other cells in the area with excitotoxicity. I like how you had those cat whiskers going across.
Yeah, no, it's cute.
That was pretty awesome. So yeah, it has to be intense, but not too intense, and repetitive, functional, moderate intensity, basically. So for let's say that you have a patient who shows up at the emergency department. Their family member reports that they started slurring their speech in the morning and were having trouble coordinating their movements, and so they brought them in. What type of problem do you think that is? Acute, subacute, or chronic, vascular, inflammatory, or degenerative?
maybe acute.
Absolutely. Do you think what vascular, degenerative, or inflammatory?
Vascular.
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 Oh, we're going vascular for sure. So sudden onset acute problems tend to be vascular. Let's say that you had someone who was in a car accident a couple days ago, and they were fine afterwards, and then they started having headache of increasing intensity. They started maybe having some blurred vision, a little bit of dizziness. What's going on there? Acute,
Subacute.
subacute, or chronic?
Subacute.
Subacute.
Subacute, definitely. And inflammatory, vascular, or degenerative?
Inflammatory.
Probably inflammatory, yeah. So they might have like a subdural hematoma, which is going to progress over time. They could have an epidural hematoma. So subdural hematoma tends to be venous and epidural tends to be arterial. We haven't got to that yet, but there it is. I'm throwing it out there. But definitely inflammatory and definitely if you can teach the cat to write, she can take the exam.
Yeah. You hear that? Learn. Learn.
Learn, kitty, learn.
He's over there. Give me
That's
that
good.
pencil.
Enough repetitive tasks, repetitive tasks.
That's good. So, let's see, we had our groups of neurons. So we have our afferent neurons going towards the central nervous system. What type of information do they tend to carry?
Sensory.
Sensory information, absolutely, up to the nervous system. What's going to go on with it? What about the efferent? What type of information do they tend to carry?
Motor.
Yep, motor. Yeah. And what's the third group besides afferent and efferent?
Interneurons.
Interneurons. And what kind of information do they carry?
Kind of local.
Yeah, local information. They're processing, they're sharing information with other neurons. A lot of the neurons in our cerebral cortex are interneurons, and they're the ones that are doing all the heavy lifting. They're doing all the processing in our spinal cord and in our central nervous system is done by interneurons, and so all of the association cortex, which is everything besides motor and sensory, basically, that's doing all the complex functions of our brain. So that's a good thing. We like those guys. So which ones tend to be the longest in terms of axon length?
Oh. Would the pseudo-unipolar?
Pseudounipolars, yeah, because they go from the periphery to the spinal cord, and the bodies of the pseudounipolars are in the dorsal horn of the spinal cord. which is where we have our gating effect. Those are like our A betas. But either the afferent or the efferent neurons, they're traveling, they're going in those tracts. So what's a tract?
Peduncle.
No. More
So
words. Use
that
your
word was
words.
stuck in my head. Peduncle.
Tract.
Um,
So tract is a bundle of axons with a common origin and common destination. So the afferent and efferent neurons are traveling in those tracts, usually along the spinal cord, up and down from the central nervous system. And the interneurons are little. They're going just short distances between this neuron and this neuron in the brain. Or in the spinal cord, just little short distances. So the afferent and the efferent neurons are long, traveling in the tracks, going long distances. So when you have a group of axons that have a common origin and common destination, it's called a tract. We'll start to talk about, when we talk about vertical systems, we'll talk about the different tracks and their names and where they go to and from. Luckily, a lot of them, their names tell you where they go to and from, like the spinothalamic tract, which goes from the spine to the thalamus. So that's nice. So we have that. What's it? Any kind of injury or abnormality in tissue called?
A lesion.
It's a
yes.
lesion. So it can be anything that is not normal in any kind of tissue. And that's not just for nervous system tissue, but you hear it a lot in nervous system terminology, like with spinal cord injuries, they'll say above the level of the lesion or below the level of the lesion, that sort of thing. When you have something in your some kind of anything that happened to your brain, that's a lesion. Any nerve, like carpal tunnel syndrome, that's a lesion on the median nerve. So if you have one single lesion that doesn't cross the midline, what's that called?
Focal.
It's focal. Yeah. What's it called if you have multiple lesions that are asymmetrical?
Multifocal.
Multifocal. What's it called if you have
Diffuse.
yeah,
Raise your
smarty.
hand, Mason.
Okay, now you got to tell us the
Last round.
definition. So, Mason, what's a diffuse lesion?
Bilateral and symmetrical.
Yeah, bilateral, maybe symmetrical, but it affects things on two sides of the midline.
It doesn't
May
cross.
not be symmetrical. Yeah. Doesn't cross the midline as a single lesion, but it affects both sides. So think of an example, a functional example of what would be a multifocal lesion.
Tuatunga biloba's
What?
concussions.
Say again, Mason?
More of a football thought, but Tuatunga biloba's multiple concussions.
Multiple concussions, sure, or multiple injuries in the same accident.
Like a motorcycle accident, car accident.
Yeah,
Yeah.
like
Yeah.
some kind of, man, you really got messed up in that wreck sort of accident. Yes. Or give an example, like a functional example of a focal lesion.
You stepped on a nail.
Stepped on a nail, sure. Or like carpal tunnel syndrome. That's a good example of a functional lesion, just carpal tunnel. Or if you have a brain infarction, if you have a CVA that's in a one localized area, that's a focal lesion. MS is like a multifocal lesion because you have plaques in a bunch of different areas in the central nervous system that are asymmetrical generally. So we have all those lesions. That's a drag. You want to do about that. But so what are the cells that are affected in MS?
Oligodendrocytes.
Yep, the oligodendrocytes. And what do those guys normally do when they're not affected?
Myelinate the central nervous system.
Yep, they myelinate the central nervous system. Which cells myelinate the peripheral nervous system?
Schwann cells.
Schwann cells, absolutely. Um, what do astrocytes do?
Regulate
Like stars.
extracellular fluid.
Yeah, absolutely. That's one of the things. They're multifunctional. That's
Clean
one of the
debris.
things they do, though. What else?
Clean.
They clean, yeah, and microglia also clean. They vacuum, they do the laundry.
Are the astrocytes the ones that kind of clean up the extra, I guess, cells that are just kind of, or was it ions that are just floating around? Or was
Yep,
that?
they do. They clean up some of the fluid. They also have a communication function. They have chemical synapses instead of the ones like you have between neurons. So they actually physically connect and they can communicate. So astrocytes are pretty handy to have around. So every neuron, so we have like all those millions of neurons, and every neuron has like its little staff of supporting cells, of glial cells. And they originally called them glial cells because they thought they were like glue that held the nervous system together, but you know that they're more than glue. They actually have function, which is good. The astrocytes also make up the blood-brain barrier. So that's pretty handy. They're keeping stuff out. And so they're making sure that we have the nutrients we need and we're not getting stuff we don't need. MS is central nervous system. It's autoimmune. It's a central demyelinating. Guillain-Barré is also autoimmune and it's peripheral demyelinating. So it affects the autoimmune function. They're not sure the exact cause. There's some suspects, but the autoimmune reaction actually affects the myelin. So it doesn't affect the Schwann cells. Schwann cells are still there, but it affects the myelin. And with MS, the autoimmune affects the oligodendrocytes. So I don't know, that seems more serious to me. And in actual fact, it is because if you have MS, it doesn't go away. If you have GBS, you can have a full recovery from it. So, because once the inflammatory process ends, then the myelin can rebuild because you still have Schwann cells, and the myelin rebuilds and you gain function and you resume your regular function, which is great. So you can fully recover from GBS. But GBS has a acute stage and a chronic stage. And during the acute stage, if your phrenic nerve is affected, then your diaphragm's not innervated and you need to be on a ventilator in order to breathe. So that's why the small percentage of people that actually die from GBS, that's usually what they die from is acute respiratory distress because of that effect. So let's see, what other lovely. So neurotransmitters, you should know for the 3G neurotransmitters, glutamate, glycine, and GABA. What's glutamate's claim to fame?
Excitatory.
It's the primary excitatory neurotransmitter in the central nervous system. And what about glycine and GABA?
Inhibitory.
Yep, they're the inhibitory neurotransmitters in the central nervous system. So the basic function of dopamine, serotonin, norepinephrine, you should know those. So what's the basic function of dopamine? It has a bunch, but you could say it in 3 or 4 words. So dopamine functions in movement control, it functions in executive function, um, cognitive function, decision making, and motivation. That's a big part of dopamine is motivation. Serotonin functions in mood and alertness. And norepinephrine functions in focus and alertness to your surroundings. So norepinephrine, when it's released by the adrenal medulla, it stimulates the sympathetic nervous system to be more alert to what's going on around you. So that hopefully makes sense. And let's see, what other neurotransmitters? Oh, the peptide ones. Um, the substance P, it's substance P for pain. It's released by inflammatory events. It's usually released by joint capsules and that sort of thing. He's really liking that pencil.
You won't leave me alone right now.
Oh.
Trying to take the test, Darren.
He goes, "Why don't you play with me? Just play with me." Very cute. But so let's see, the substance P. What else? What else do we have for neurotransmitters? There are like a lot of other neurotransmitters that we didn't really even talk about. So there are ones that are involved in sleep, and there are ones that are involved in, like everything we do, like every single little thing we do. But the
Is
main
acetylcholine on there?
acetylcholine, that's who I was forgetting. So what's acetylcholine's claim to fame?
Muscle.
Yeah, it's the one that functions at the neuromuscular junction. So when acetylcholine is released at the neuromuscular junction, it is always excitatory. Always. You never have an inhibitory signal to a muscle. It's always an excitatory function. Acetylcholine is also a heavy hitter in the autonomic nervous system. So, and it can be inhibitory or excitatory depending on where it binds. But at the neuromuscular junction, it's always excitatory, always stimulates a muscle contraction, which is what we want. So we'll just have like cat hour. I like it.
I only have Darren's thing up just so I can watch the cat right now.
Yeah, that's reasonable, I think. So acetylcholine. So the different types of receptor ion channels have different stimuli that cause them to open, the different ion channels. So what is the stimulus that causes a voltage gated channel to open?
Sodium?
Well, movement of sodium, definitely. So you have a local potential that changes the membrane potential at that area, and that will cause voltage gated channels to open. What for a modality gated channel, what's stimulus?
Touch, chemicals.
Right. Pressure, sensory information, basically. And so what about a ligand gated channel?
It's key.
So something has to bind to a ligand gated channel. So that's our synaptic receptors. So that basically, if you think of it in terms of let's send a sensory signal up to the brain, you stimulate a modality gated channel with pressure or whatever, light touch, and then that sends the action potential to the next neuron in the chain, which stimulates the voltage gated channels to open. And the synaptic vesicles go across, release neurotransmitter, binds, which stimulates ligand gated channels to open and propagates the action potential. So they can all be functional in one route of one loop. They call them loops when you get a bunch of neurons working together. But each one kind of has its little part to play. What do leak channels do?
They maintain balance.
Right, they allow mostly their chloride. They allow chloride to go down its concentration gradient, and let negative ions into the cell to keep the inside of the cell more negative because chloride is negative. So the leak channels like the drip irrigation. There's just like it's constantly open. There's just a little bit going through all the time, and the other channels are like the sprinkler system that's on a timer. It opens and then it's watering. But or you live in Western Washington, you don't water stuff.
Or North
Nature
North
does it
Coast
for you.
Oregon.
Yeah, or on the coast of Oregon. So okay, so how's everybody feeling about all that stuff?
I think you just telling us that kind of gives a little bit more direction of like I know some of this, but not all of it,
Mm-hmm.
or I need to revisit it.
Yeah. So that's a good, I mean, really that's the information that we're dealing with right now. And if any of that feels like, wow, I had no idea what she's talking about, then you should probably look that stuff up.
Kind of how I
And
feel about a lot of the multiple choice stuff is that, when you have four examples to look at, you can kind of cross eliminate, and then you're like, oh, you pull in information back from the back of the brain. You're like, okay, this is starting to make a little bit more sense. But just having to recall specific details like this, I'm struggling a bit with it.
Said,
that's legitimate. But it's good for you too. When you're struggling, the struggle is good quality work. If everything's easy, then you're not really learning anything. If you're struggling, this is good. So the struggle is of value. So don't feel bad if you feel like you're struggling. But practicing, that's what I always say. Taking exams is recall. And you only get good at recall if you practice recalling things. And so quizzing yourself or having someone else quiz you is really a useful thing because you practice that recall. So take even just taking the learning objectives and pretend that you're me and you have to make up test questions about this, make up test questions about those learning objectives, and then send them to somebody else and say, hey, I made these test questions. Take the test and see what you get on it or something like that. That's helpful. Or even just explaining it to somebody. So pick your unsuspecting friend or family member and say, hey, can I talk to you about the events at the synapse?
My family would tell
And
me no.
well, yeah. So you can't do it as a question. You go, let me tell you about the events at the synapse. And if they go, Tony, you're insane. And you go, no, this is going to help me. So just bear with me.
What you
Usually turns into them just looking completely blank-faced at you the
And
whole
that's
time.
okay. That's okay. That's
What
like
you do in the
I would always
teaching
tell my mom stuff when I was in school, and she'd go, well, I don't know what you're talking about, but it sounds good to me. One time I sent her a Mother's Day card that said, Happy Mother's Day, thanks for all the mitochondria, because you get all your mitochondria from your mom. You don't get any from your dad. And it had cute little animated mitochondria on it. And she goes, well, that was pretty cute. I don't know what you're talking about, but it was cute. So
Better yet, when a
sometimes you have to
patient
bore
checks in
your
early.
family.
Or you just let a patient check in early, just talk to them for 10 minutes straight about it.
Sure, that's a great idea. Or
And they
ask,
regret
say,
showing up.
hey, do you have any questions about your injury? And then they can ask you and you can regale them with all the information you're learning.
So if a patient won't leave the front desk, just start telling them the stories, and that should work.
And they go, I gotta make some phone calls before. All right. Any questions before we sign off for tonight?
I don't think so. I feel like I have enough to kind of review. So
Okay. That's good. So in the last couple of days before the exam, you should be kind of just wrapping things up, getting your cheat sheet together, just like a final review. The night before the exam, don't stay up real late studying. Just have a nice meal, do a good workout, get some good sleep, and then you'll be ready for the exam. Maybe a light review. Sound like a plan?
Or
Yep.
don't sleep at all, lots of caffeine.
Mason,
worry about you sometimes. Did you hit your
The
head when you
wheelchair-
got out of the wheelchair?
The wheelchair was fine. Ryan and Christian can attest to that. I moved around great.
Wheelie champion.
May or may not have went into traffic
may or may not.
a few times. So
You guys
That's
were
true.
in traffic. Were you not?
I was
He
keeping up.
legitimately went through the roundabout.
Yeah, I
That's
see
what we
there's
were
a
sent
guy
to
that
do.
must live in the assisted living facility over there that I always see him in his power chair going through the traffic circle.
Tony almost killed Sophia. That's okay.
No, I didn't almost kill her, but she did fall out of her wheelchair.
Okay,
almost killed
so
Josh.
that's what I say. It's one of the nevers of physical therapy. Never fall because there's too much paperwork involved.
Yeah, well, I'm glad it happened to one of us students and not to a patient.
Exactly. So that's sometimes a lab is the time to learn what not to do.
Oh, I definitely learned that.
Okay, that's good.
You said that really quick there, Tony.
Oh, yeah. Not my
Well,
greatest
that's
of days.
There's a group in Bellingham that through like the Parks and Rec that it's a group of people who use wheelchairs for mobility and able-bodied people. It's a big group. They go and play murder ball, and everybody's welcome. You can just go and play, and it really is fun. And it's a good workout too. Like zooming around in those wheelchairs, trying to catch the silly ball. Yeah.
I'm gonna have to look up what murder ball is now.
Oh, look it up and watch. There's a movie. Watch the movie. It's wheelchair rugby. It's a Paralympic sport.
I was just going to ask if it was a version of rugby.
It is. It's wheelchair rugby. There's a movie called Murderball that you should watch. It's great.
There actually is.
Look it up. And it's been out long enough that I'm sure you can find it free somewhere.
Yeah.
2005.
2005.
All right.
So, yeah, it's really good. All right. See you guys on Thursday.
Sounds good.
All right,
Thank you
see
so
you
much.
then. Thank you
See you
again.
then. Thank
Good
Don't
night.
stay up too late.
Yeah.
We have another test to take anyways, so
Woohoo! Yeah.
Mhm.
Go
yeah.
take that test.
Yeah. All right. See you
Bye.
guys.
All right.
See ya.
Bye.
Thank you.